And what I'd like to do here today is give you an idea of what we do with GWAS data and what new functions we've added recently to enhance the environment for looking at GWAS data. And I'll go into a project that already has some data imported, although it hasn't had much work done on it. And let's talk a little bit about what the data looks like. So we already saw the spreadsheets with the sequencing data. And here we have essentially the same format, where we have all of our SNP data in columns. There is this embedded marker map feature that goes with it, and then we have all of our samples in rows. This is a relatively small data set that we're looking at here today. It has 565 samples, and it comes from the old AFI 500K platform. So we've got about 500,000 SNPs. And then I also have a spreadsheet with some basic phenotype data in it. And the first thing I need to do, I forgot about this here, but SVS is able to work with all different types of phenotypic data. Here we see the letter B indicates a binary variable, C indicates categorical, R is real value numeric, I is integer. Typically, though, if you have a binary trait you're looking at, we do prefer to have it coded as binary. And SVS does have the ability to edit data on the fly when necessary. You do have to go into a separate spreadsheet view mode. So here, if I want to convert this to a binary representation, I can do that in the editor. If there are individual values that I know are incorrectly entered that I need to change, I can do that in the editor. There are also options for some basic text manipulations like splitting columns on delimiters, appending and concatenating columns, and so on. I'll go ahead and save that change, and then merge my phenotypes and genotypes, and take a look at what we're able to do here with this data. What I'd like to do in the time that we have is go from this spreadsheet full of SNP data with a few phenotypes and get to the point where we can run a mixed linear model association test across the genome for this set of data. Now, the interesting part about using mixed model analysis in GWAS is that the mixed model tools that we incorporate use a kinship matrix to explicitly account for the pairwise relatedness between all sample pairs. And because of that, what we'll need to do as a workflow is start by selecting an informative subset of SNPs, calculate a kinship matrix, and then proceed with our mixed model analysis. So to begin here, I would like to apply some filters based on call rate and allele frequency. And in the genotype menu, you'll find a lot of the functions that are most useful for working with SNP data. So genotype statistics by marker is going to calculate allele frequencies, call rates, Hardy-Weinberg equilibrium. Genotype filtering by marker is essentially the same calculations, but it allows you to create subsets based on thresholds. Statistics by sample is going to calculate sample call rates, autosomal heterozygosity, gender inference based on the X chromosome, and so on. But we'll go ahead and start with filtering by marker. And I'm going to just drop out any SNP with a call rate below 95% and an allele frequency below 5%. So I'll just take some pretty basic filters here to start with and click Run. This should go fairly quickly. When this is done, the next thing that I want to do, well, first of all, let's take a look at the output. You'll notice here we get this filtering results where we see the call rates, the allele frequencies, all laid out. There's a flag indicating which SNPs should have been filtered out. If I want to visualize any of this, it's just 2 clicks to get a histogram of the call rate. If I want to explore this lower tail, see just how many SNPs are down in this low call rate region, I can, but I'm comfortable that most of my data has pretty high call rates. But you see back here in the background, whoops, didn't mean to do that. Just like we saw with the sequence data when we applied filters, here with the SNP data, we see columns that have been turned gray if they failed either the call rate or allele frequency. And just like with the sequence data, I can click the subset button to reduce just to the data that's important, or that passed the filters, that is. So now, if I want to create an informative subset here where I have SNPs that are not correlated with each other, there's a function back in the genotype menu under quality assurance for LD pruning. I'm going to adjust the settings here to go a little faster. The defaults are usually fine. But the way I set it up is that it is scanning the length of the genome using a moving window of 30 SNPs at a time. And within that window, if it finds any pairs of SNPs that are strongly correlated, it's going to keep the first one, drop the second. And I suspect that when it's done, we'll end up with about 36,000 SNPs, which will then serve as the basis for calculating a kinship matrix that we can then put into our mixed model analysis. But I think we might also pause to do a quick principal components scan on this data to see if there's anything there that we need to be aware of. So as expected, we ended up with 36,000 or so SNPs. And I'm going to rename this spreadsheet, call it LD pruned. Let's talk about what we can do with this particular data set now. I have in the project here in the background this HapMap reference data from the same chip. It's also from the AFI 500K. If I want to combine those so that I can look at population stratification, it's a pretty simple process. Just click the append button. We'll bring it together. We'll just get that same set of SNPs, so we'll drop any unmatched columns. This should go relatively quickly. And now we have all of those samples in one big spreadsheet. So I can look at the principal components here. And I'll go ahead and run this with the default settings. Principal components is one of the functions in SVS that is multi-threaded. I'm working on an 8-core CPU here, so it's able to take advantage of that to speed things up. And when this is done, we should be able to visualize this data a little bit and get some idea of the population structure that is present in this data set. So there we go, that wrapped up pretty quickly. We get two outputs from principal components analysis. One is just the listing of eigenvalues. We can see that the first two are very large compared to the remainder. And then we also get this spreadsheet of the actual principal component matrices, or vectors, excuse me. And I'd like to merge that with the phenotype data just so I can get everything in one place here. And then in SVS, we have the ability to visualize any variables, really, in a scatter plot. And as we look at this, we see this sort of classic triangular pattern that you often see with SNP principal components. And I can recolor it based on the population data from the phenotype table and see here very quickly what we have are the Asian HapMap reference group up here in green, the European HapMap reference group back here in blue, the African samples over here in the gray, and all of my study samples in orange. So clearly we have some pretty significant stratification in this data set, and we'll need to be aware of that as we go forward. If you want to drop samples based on these patterns, there are options for outlier removal based on principal components analysis. I won't go into that too much here today. But also, The mixed model analysis that we're using is really quite good at accounting for population structure and stratification. So we'll see how that works out. So the next thing I want to do here is actually calculate that kinship matrix that we're going to need. And that's going to be, again, in the genotype menu under quality assurance. There's this tool for IBD estimation. And if anybody is familiar with Plink, the method that we're using here is very similar to the, I think it's the minus genome option in Plink. And we can output a matrix of IBS distances, a matrix of pi values, or just a pairwise data set where we can see all of the pairs above a certain threshold. We'll go ahead and let this run. I think this one may take just a bit longer. But maybe while it's running, we can check and see if there are any questions that have come in. And one of the common questions that we see a lot is in regards to file formats and import formats. And I haven't emphasized that very much here today. But if you have GWAS data, we can read from all of the standard formats that get used in the open source community. So if you have PED files or if you have Plink formatted files, we can read those directly and convert them immediately into our internal spreadsheet formats. If you have data from Illumina in one of their standard formats, like the final report, we can read those. We also have our own plugin that works with the Illumina Genome Studio software. So if you're using Genome Studio to get your variants or your genotype calls, you can use that plugin to export the data out of Genome Studio directly into our preferred formats. Then also, we support import from a variety of other what we call third-party formats. But if you have data that's in Excel or in SPSS or SAS or other statistics programs, we can usually read those directly as well. So I'll also point out that pretty much any format that we can read, we can also write to. And if you just need a software tool that allows you to read a VCF file and then export something in Excel, for example, you could use SVS for that. Obviously, there's a lot more functionality beyond just reading and writing different file types, but that option is there. Okay, so the IBD analysis finished, and we see we have this listing of pairwise IBD estimates just for the ones that look the most related. We also have this matrix of estimated pi, similar to the pi hat statistic that you would get from Plink. And this is a complete matrix with all pairs in it. And what I like to do with this is just visualize it. We can throw this into a heat map type visualization. Maybe I'll adjust the coloring a little bit so that we'll make unrelated people white, the stronger relationships, let's make red. And we could also throw in, just for fun, an intermediate yellow color and change the range on that. So here we should be able to see any unexpected relationships pop out, but also down the axis. This data was previously sorted by family. There are a few families that are known to be in this data set. And so if we come in right here, this is a family trio where we have two parents that show minimal relationship to each other, so this pi value of 0.04, while they are both first-degree relatives to the child. So we see the value of about 50%, which is what we expect with the first-degree relative pairs. And if we zoom out and look around, we can see different types of structures in here, but for the most part, everyone should be either unrelated or part of a trio, and there are a few scattered trios in here. So now that we've done that, we've gone through a relatively thorough workup on GWAS quality. There's obviously a lot more things we could look at. We could get into heterozygosity, we could get into inbreeding coefficients, different directions we could go. But we've looked at the principal components down here to see population structure. We've looked at pairwise relatedness. And what I'd like to do now is come back and just run an association test. And I'm going to the spreadsheet where we had all of the SNPs that were above our frequency threshold and minor or call rate and frequency threshold. And I have several different phenotypes here that I might want to test for. But I think the one that I'll use today is this blood pressure phenotype. And this is all simulated data, by the way. But if I just want to do a simple, naive association test, you can run the association test function. You have the option of several different genetic models. With each model, there are different statistics that make sense for it. I'm using a quantitative dependent, so I'm only getting tests that work with a quantitative. If you are using a binary, then you'll get other options here like chi-squares and Fisher's exact tests and Cochrane-Armitage tests. But I'll go ahead and run this additive model and get a few extra outputs here, so you can get things like call rate and allele frequency output side by side with your p-values if you want. So we'll let this run. It's going pretty quick. We're doing about 250,000 association tests in roughly 10 seconds here. So with our results now, we see we have p-values for every SNP. We have test statistic, expected values for the p and for the test statistic. We can scroll through and see all of these other values here side by side, like allele frequencies. Now, if I sort by p-value, I can see that we have some signals around about 10 to the minus 8th, roughly. And looking at those, I can also come through and get an idea what the allele frequencies are. If I had case control data, it would be broken out by case control status. There are also options when you have quantitative traits like this to get average values of the quantitative trait for each genotype group and so on. I didn't turn that option on here. But of course, what a lot of people like to do is just visualize this data. We've already seen the visualization interface when we were working with the sequence data. But here now, I can use it to look at the SNP data. So I can see I have this pretty strong peak coming out on chromosome 16. And if we zoom in over here, this is interesting. We're coming in on the FTO gene. And again, I'm telling you this is all simulated data. But FTO, if we come in and learn about it, I can click here and I get these links out to external databases. But I'm sure you're probably familiar. FTO is actually an obesity gene. I don't know why it should be related to blood pressure necessarily. But let's talk about how we might account for obesity in our test for blood pressure. Perhaps there's some confounding here, and that's where I want to get into the mixed model tool. And the mixed model analysis down here at the bottom of the menu is one of our newest functions for working with GWAS data. The method that we've adopted is based on the EMMAX or EMMAX method. And as I mentioned, it uses a kinship matrix to account explicitly for the pairwise relatedness between sample pairs. It's able to do a pretty good job of accounting for population structure at the same time. I could, if I wanted to here, I could also account for principal components as a fixed effect. I won't do that today, but I will include BMI as a fixed effect, just to see what happens when we adjust for that. And I think there we're ready to go. So we're going to do a mixed model GWAS based on the MX method, using the IBS distance as a placeholder for the Kinship Matrix. And let's make sure we're getting all of the outputs we want. This looks good. We'll go ahead and let it run. So this is a two-step process. Hopefully it won't take too long to run. I didn't time this ahead of time to see what would happen. But while we're waiting for it, I think I will just open another instance of the program and come in where I've got some work done on the same data set. So to give you an idea of some of the other things that can be done here, this is an example based on one of the other phenotypes that was in the data set. When you run a naive GWAS, and this one, I'm trying to remember what we had. So this was one where we had some pretty significant inflation, due partly to the population stratification that we saw in the data, partly maybe due to the fact that we had some families in there that we weren't accounting for. It had a lambda score. I think we calculated the lambda on this one right there of 1.8. So that's some pretty significant inflation of significance. If we did something like a lambda correction, a genomic control correction on that, we're able to bring everything in control, although we end up overcorrecting, I think, some of the more significant results. If we take that same exact phenotype and run the regression test, linear regression with just a PCA correction using the principal components as fixed effects, that also does a pretty good job of accounting for it. But then the mixed model, the MX, if we look at that one, and it, I think, does a little bit better job. It kind of cleans up the distribution here a little. But if you look at what happens with all three of them together, this is kind of a big messy picture. This was a phenotype that wasn't simulated to be associated to any particular locus. It was actually simulated to be associated with the population structure. But what you can see is in green are the results from the naive GWAS, and there's just a lot of inflation. You can see that in the background, the values are just quite high. When you use the PCA adjustment or the MX MLM, the mixed model test, it really pulls that down. We could see that with the QQ plots that we were looking at, but here maybe it's emphasized a little bit more exactly what is happening. Now, down here at the bottom, this is an example of how you can use different types of annotations with GWAS data. Suppose we were interested in this little feature peak right here. Let's zoom in closer on it. What I have here is, actually this isn't the one I thought it was, but it was one data set that I was playing with a while back to represent recombination hotspots, and so just so you can get an idea of what the limits are of the conserved region around the peak. And so you can kind of see that here. This is from Decode. There are other sources you can get from HapMap that show the same thing. Then also with SVS, we have the ability to view and interact with LD plots. So if you need to explore the regions around your significant results, see what the LD structure looks like, click around and see the actual strength of the relationship between pairs of SNPs. That is also fully supported. So let's check back and see how our progress is going. So that mixed model test is still running here in the background. It's going to take a few more minutes to finish, but I think we've shown pretty well what it does looking here in this other project example.